Hello, this is Module 2, Section 2. Good thing that I can't keep track of numbers. Love that for me. Okay. Take 2. Here we go. Hello, this is Module 2, Section 2. In this lesson, we will be reviewing synaptic transmission. Which is exciting because we've spent a lot of time on introducing the action potential, talking about electricity in the brain, talking about electrochemical gradients, and finally we get to put everything together in synaptic transmission. So we know that the neuron communicates through conduction of electric action potentials that are driven by the electrochemical gradient. So it's differing concentrations of ions all around the axonema that move, and they cause this electrical impulse. Our key questions for today are what happens after the action potential. We're going to formally introduce and talk about neurotransmitters, and we're going to talk about the kinds of receptors found on dendrites. So synaptic transmission as a whole refers to the process from signal binding at the dendrites to neurotransmitter release at the synapse. By signal binding, I mean neurotransmitters binding to receptors on the dendrites. Depending on the type of neuron or the type of channel, signals can be inhibitory or excitatory. Excitatory transmissions will depolarize the membrane, causing an action potential, and inhibitory transmissions will hyperpolarize the membrane and prevent an action potential. So this is a quick schematic of the synapse. So here we see when the action potential makes its way down to the synapse, it opens a voltage-gated calcium channel, which then binds to synaptic vesicles and transports them to these docking proteins. Now, a special characteristic of synaptic vesicles is that they can fuse with the membrane, which causes a release of the contents inside the synaptic vesicles, which, of course, those contents are neurotransmitters. The neurotransmitters move across the synapse and they bind to receptors on the dendrites. So, what types of receptors are there? There are ionotropic receptors and metabotropic receptors. Ionotropic receptors we've actually talked about before. These are chemically gated ion channels, okay? They're typically associated with really quick transmissions because if it's a sodium channel, the neurotransmitter binds, the channel opens, and of course sodium rushes into the cell and it immediately depolarizes the membrane. We can have inhibitory ionotropic receptors. This just means it's selective for an ion other than sodium, probably a negative ion, to hyperpolarize the membrane. Metabotropic receptors are a bit more complicated. They basically cause a series of events inside the cell that eventually cause an action potential. So of course these are longer transmissions, and they can be excitatory or inhibitory, just like ionotropic receptors. For a metabotropic receptor, the action potential is a bit more of an indirect consequence of these intercellular events, okay? So just a closer look at ionotropic receptors. As we can see, the neurotransmitter will bind to a spot on the ion channel, and the ions can move through. Pretty simple. When the receptor is not bound, ions cannot move through. Metabotropic receptors, like I said, more complicated. So another name for this are G-protein coupled receptors. And G-protein coupled receptors in general are found all over the body. But basically what happens is the neurotransmitter binds, which causes a dissociation of this part of the channel, which then activates an enzyme and causes all of these secondary events, activation of a secondary messenger, and other cellular responses. This can include phosphorylation or activation of other enzymes and stuff like that. And then eventually this will open or close ion channels associated with the action potential. So if it is an excitatory transmission, we get binding at the G-protein coupled receptor, dissociation of the protein here, and all of our secondary events, and then eventually the channels open for sodium. But it's just not as direct as ionotropic receptors. So in summary, we're about to look at the excitatory transmission process as a whole. So the receptor binds on the postsynaptic membrane to either ionotropic or metabotropic receptors, and then the membrane is depolarized. Since this is an excitatory transmission, we're assuming that the action potential occurs. So of course the membrane is depolarized when we have an influx of sodium cells. The depolarization conducts all the way along the axon. When the action potential reaches the synapse, it opens voltage-gated calcium channels, which transport those synaptic vesicles to the edge of the synapse, and the neurotransmitters are released into the synapse, and they bind to the postsynaptic membrane. So now we'll talk about neurotransmitters, finally. It's been brought up before, but these are the chemical molecules that act as messengers in synaptic transmission. So their function is to cause or inhibit action potentials. So the types that we have biogenic amines, amino acids, peptides, purines, gases, and lipids. And there are so many different neurotransmitters. Of course, we've identified some really important ones, such as dopamine or serotonin, which are both associated with mood regulation. Dopamine is associated with motor regulation as well, but we are nowhere even close to knowing all there is to know about neurotransmitters, so lots of work to be done there. We're going to now look at this whole process in this one little diagram. So neurotransmission starts with the synthesis of neurotransmitters and formation of vesicles. So this is happening in the soma through the endoplasmic reticulum and the Golgi apparatus. Then these vesicles are transported down to the synapse through axoplasmic transport. Then we have the action potential. It travels down the axon, and once it reaches the synapse, we have opening of calcium channels. These calcium channels assist the transport of the vesicles to the edge of the membrane, which then release our neurotransmitters. These neurotransmitters move across the synapse and bind to receptors on the postsynaptic membrane. Then we have this process called reuptake, which is actually really important. And this either means literally the neurotransmitter is taken back up into the synapse that it came from, or it can just mean breakdown of the neurotransmitter in the synapse by an enzyme. So the process of reuptake is actually really, really important, because if we have neurotransmitters hanging out in the synapse, they can just constantly stimulate the next neuron, which is not something we want. We want to be able to shut off signals when we need to. But sometimes we need to block these reuptake transporters, right? So people who have depression often have lower levels of serotonin. Why? We don't exactly know, but they do have lower levels of serotonin. And so what some drugs do are they're called selective serotonin reuptake inhibitors. So they block the reuptake transporters, which means that the serotonin has more time to hang out in the synapse and stimulate the next neuron, which causes those, happier feelings. So reuptake is actually a really, really important process. We also have another example is acetylcholine. So acetylcholine is found at the neuromuscular junction. It's associated with activation of muscles, and the enzyme that breaks it down is acetylcholine esterase. And if acetylcholine esterase doesn't do its job, then our muscles are continuously stimulated, and this causes muscle spasms. So once again, just demonstrating the importance of this process. So quick summary of this lesson. We know that the neuron is the main functional unit of the nervous system and is responsible for carrying neural signals. And this is achieved through reception of a signal at the dendrites, conduction of the signal through the axon, and release of neurotransmitters at the synapse. So next, we will be stepping back a little bit and looking at sensory systems.